Hi, Aaron. Hi, guys. All right. We'll get started very shortly. And then if you can take a look at the question. Aaron, have I taught you before?
No.
No. Okay. Hi. Okay. Thank you for turning on the camera. Yeah. Okay. So we're going to do, well, this is test number 7, which is a free response. And we'll wait a little bit, but so I'll keep this one open here. Test number 7. Okay. All right. So, and this is the equation sheet. Let me get it going. All righty. Okay. So we'll get started in a little bit. Okay. How about you guys? Did you guys do the test? And then, yeah, no, didn't get a chance this week. Okay. So then just kind of look over the questions and then we'll see how you guys score. This is free response FRQ. So if you recall, the questions are for the new test is a little bit shorter than this one. So the AP Bio, hi Michaela. So AP Bio here is going to be shorter. So I'm going to actually move this so I can actually have enough room. Thank you. And then here, so the first 2 questions are going to be 10 points in case we constantly review this because we may be stressed for time. And these 2 10-point questions will involve, one of them will be graphing. Hi, come in. So one of them will be graphing, okay, and experiment. Hello. All right, so let's make sure we understand this. So we are pressing towards the AP test very soon. So make sure we are accustomed to these kind of questions. Last week we did a multiple choice together. Okay, I think most people felt the multiple choice was not as hard as the free response, and that should be the case. Yeah, free response, regardless, is a little bit harder. It's just a little bit harder. Yeah. So again, the free response, the first 2 questions are 10 points each. You're going to be dealing with experiment, graph, and data. So those are the majority of the point. Now, 3, 4, 5, 6, those are 4 points each. So you can see that 4 point times 4 is 16, and that's 20, right? So that's the new test. The old test, okay, the participant can now see your screen. Okay, I was hoping they would see my screen a while ago. Okay, so anyway, I'm going to close this. So the older test that we are doing right now, there's also 7 and 8. Okay, so these are 3 points each. Okay, so we're gonna the old questions are, yeah, I can combine them. So the old tests right now that we're practicing with are similar. I think the newer tests are a little bit longer because they shorten the test number. So question 1, 2, 3, 4, 5, and this is 6, 7, 8. So these are 3 points, these are 4 points, and these are 10 points. Yeah. So this would be the old test. All right. So that's the one we're practicing with. So they're actually nice enough to shorten this. And then let's see how that goes. So what we're going to do is as we go over every question, please add up your points. Okay. So if you have the points total together, then we can finally we can add it together. Yeah. So at the end, so they can give it to office how you do. Okay. So let's look at this first question. Okay. So this is question number 1. Okay. All right. The question is about whether the timing changes for AP. Yeah, for the second part, it's still 90 minutes, but it's now over 6 questions. So they will tell you the time. It's actually distributed evenly with the points. So we'll work on the timing. I don't think, well, the old test is hard to finish with 8 questions. People get very stressed out, and there's no penalty, so you want to try to push as fast as you can. So the old test, I would assume is a little bit easier. It started last year. So most students out okay with the time, but they worded so weirdly that some of the concept that is actually not too difficult become actually difficult because the wording. Yeah, so you have to get around the wording. So if you have good reading skill and writing skill, that is definitely the most important skill here, answering full response. And of course, you have to analyze this. Okay. All right, let's look at this. So I'm going to switch between drawing on the screen and also writing out notes. Okay. So again, this is recorded. So once we've after the class, this will be posted. So if you need to go over specific mistake concept again, you can just download the recording and fast forward and skip all the part I blab about other things. Okay, so let's look at this one. So as you go over this, you have to make sure you highlight selectively. So it's a survival genetically modified. And now this one, you don't have to attempt to pronounce this. I just skip it altogether. I don't even look at it. So it's BT, right? So I use the first two letters. So you have BT strand, okay? So this is the BT strand, okay? That's a bacteria. So they tell you background information, which is important for answering the question, okay? So here it says, in bacteria community, where resources are often limited, survival requires the ability to sense, ability to respond, and ability to cooperate or compete. So these are all the different interactions. We already know this, they don't have to tell you, but this is a good reminder, right? You have the sensitive environment and different populations, they compete for resources, they can work together, they can prey on each other, right? In a community containing, this is the BT, okay? These abilities rely in part on contact-dependent communication. So that's the whole point of this, right? With neighboring cells. This communication involves a signaling protein, protein S. So we introduce this new protein that gets transported to the surface of the cell. And when in direct physical contact with another bacteria, the S is cleaved and internalized by the recipient. So that's a lot of information in this sentence. So definitely that's the sentence you want to focus on, right? So again, to the surface of the cell, direct physical contact with another, and it's clear by the it actively okay, nuclease breaks down nucleic acid. So if you repeat and paraphrase, it will help you understand. So that the contact-dependent communication requires protein S, which is put it to the surface of the bacteria, and then when it's in physical contact with another, that's the contact dependent, right? And S is cut, internalized by the recipient cell, somebody else, right? Not you. You produce the S and is internalized by somebody else. And then it will be a nucleus. A nucleus will break down the nucleic acid. So I guess kill the other cell, okay, if they're too close in contact. There are different forms of protein S, S1, 2, 3, and different forms of internal protein, okay, R. So R1, 2, 3. Recipient cells are protected from the nucleus activity of protein S if they produce the appropriate form of protein R. So what is the difference between S and R? Now the S is to attack, right? And you have different forms of protein R, R1, 2, 3, so they're protected from the nucleus activity if they have the correct protein R. So R is to protect, S is to attack. Okay, so you can see that we have three different cultures here. We have S1, S1, S1. They all produce S1, but here's the protein R. Culture 1 does not produce protein R. Culture 2 produces R1. Culture 3 produces R2. Okay, so that's what we are told. All right. Okay, so let's look at the next part. So in an investigation of the BT strand were genetically engineered to produce different combination of S and R. The cells, so this is the paragraph that talk about the table, right? The cells were placed in a nutrient deficiency medium. So there's no, why would you think they want to put into a nutrient deficient medium and culture for 4 hours? So the idea is that this, without nutrient, this will induce some kind of competition, I would assume. Okay, in culture for 4 hours, the density of live cells in the culture was recorded at the two time points, 0 and 4. So they're measuring how much cells produced, T0 and T equal to 4. Okay, so, all right, so that's what this is, right? Okay, so if you look at the result at T0 and T equal to 4, if you don't have protein R right here, and your cell density cell density dropped, right? Okay, like that. So that's kind of what they're doing here. Okay, so the question is, construct an appropriate labeled graph that represent the density of live cells in each culture of the 3 genetically altered BT strands at both time points. So if you look at the structure of the points here, usually a bold term is 1 point, but when they ask you to construct a graph, there's more than one. So if you look at part B, explain is 1 point, describe is 1 point, right? But this is for 2 and 3. Usually putting together a graph is like 3 points. And this may be a 1 point here, right? Describe the effect of expressing combination of S and P on 2 and 3. So maybe 2 and 3, so that's 2 points here. So that would be 6 points here, and this would be the 7. Maybe this is 8 and 9. Yeah, so I'm just guessing the points here. Okay, and on the new test, again, the free response is going to be either 8 or 8 to 10. That's what the College Board says. So I'm going to assume it's 10, but it does say it is actually 8 to 10 points. Yeah. So if you take the higher range, it will be 10 points. Okay. All right. So we're going to do this graph. So let's take a minute on this graph. I should have done this first. Okay. So let's. I'm just going to take a picture of this, and then if you haven't done so, you should start thinking about every time you look at the graph, you want to know what your X and Y axis is. And you want to know the scale, right? It cannot be too small. It has to at least cover a good portion, okay, of the graph paper. Okay, so I have to cover a good portion. And then you have to decide whether you use a scatter plot or bar graph. In this case, it will be a bar graph. Yeah, most of the graphs that we've been working with are going to be bar graph. Okay, so here, okay, so the y-axis would be the density. So try to use the same wording. So don't just put density here. If the wording says density of live cells, then you write down the density of live cells. And you have to put units, otherwise it's wrong. So it's a log of whatever the CFU is, okay, and then per mil. Okay, so you don't have to know what CFU stands for. And then on the x-axis would be the cultures, right? You have culture 1. So this is the culture number. Culture 1, 2, and 3. But each one you ask to graph 0 and 4, right? So construct label the graph of the live density. Density of live cell in 3 genetic culture at both time points. So T0 and T4. So that's why bar graph was nice. So let's look at the x-axis. So it looks like the density goes all the way to about almost 8, right? So 1, 2, so maybe like 1, 2, 3, 4, 5, 6, 7, 8, 9, right? So like that. Okay. So and make sure you label the axis after it's done. And the x-axis is the 3 cultures, 0 and 4. So let's graph culture 1 at 0, 7.3. So you can see 7.3 is probably going to be around here, right? So how wide does your graph have to be is up to you. So I'm going to use this, right? So this would be the result. Okay, I can't make it straight. I have to do this. Alrighty, like that. Okay, so there is no error bars to graph. I think if you do get it, you're gonna have to graph the error bar, the standard error, the mean. So make sure you know how to graph that. And then next one is at 4. So if you look at 4, it's 4.5. So that's 5. So 4.5 will be here. Okay, so also I'm gonna use two intervals. So like that, right? Okay, so this would be t equal to 0, and that would be t equal to 4. So t equal to 0, t equal to 4 hours. Okay, you can shade one of them to make them different if you want. Okay, and then the next, so this is culture number 1, right? And culture number 2, you do the same, 7.9. So it's like almost 8. And the other one is 7.9. So there's no difference here. So it feels like that for this culture, okay, R1 does a good job, right? Remember the R1 protein protects it? It seems to protect it pretty well, right? Okay, so, and then, so this again, T equal to 0, T equal to 4, and this is culture number 2, right? And then you can shade likewise, we're shading the final density. And 6.5 to 3.8, that will be the last one. So 6.5, maybe around here. This is 7, 6.5. And then 3.8, so almost 4. Right, so maybe around here. And then again, you construct this, right? So again, the final one we shaded. And this one we don't, right? So T equal to 0, T equal to 4. So this would be the basically the answer, right? Okay. Let me see. Okay. Okay. So I'm gonna text office for them to bring something to me. She forgot. So there it is. Okay, so that's there. So this would be like 3 points. Yeah. So if you have done this, can you give yourself 1, 2, or 3 points? So of course, you have to plot it correctly. That's 1 point. There's no standard error to mean here to plot, so there's no error bar. And the units would be another point. So make sure you have the X and Y axis label. You don't need a table. Now, if you really want to key, that is fine, because I actually labeled on the bottom, but if you want to key, that's fine. It's not necessary. And the third one is, the third point here is just, you have, okay, everything is to scale, right? So you can't just use a fraction of the picture. So if you draw this to relative scale, and you also have the units and the graph, and that will be 3 points. So you can give yourself, okay, out of 3 points, please. Okay, so that will be part A. Let's look at part B. Explain the effect of expressing only S1 on culture 1. So this would be like a data analysis. We're saying that, okay, look at culture 1. So describe the effect. What is the effect of expressing S1 only? Well, describe the effect of expressing, so this would be just part B, yeah? So how would you answer part B, huh? So remember, you have to convince yourself before you start typing it out, right? So what is that expressing S1 only? If you express S1 only, which means you don't have the R protein to protect, okay, you're actually, it's actually detrimental to the bacterial culture, right? You can see that from the cells. The cell density dropped significantly, right? So you want to quote that and say that the cell density, so how to explain, right? Okay. So a lot of people just put down expressing of S1 only decreases cell density from 7.3 to 4.5. Now, this will not give you points. Can you see why this is not a good answer for this question? Yeah. If you look at the question again, what they're asking is not to state what had happened, right? They want an explanation. So there is no explanation if I simply put like this, right? So if I put like this is not a good answer. This is wasted time. You don't have to tell them. You have to kind of explain it, right? Okay. So the explanation is that without the R protein, okay, so it kind of tells you here, right? So the R protein, the recipient cells are protected from the R protein from the nucleus. Nucleus is S, right? So the R protein protects the cell from the S nuclease if they produce the appropriate number form of R, right? Okay. Okay. It's not in the folder. Okay. That's okay then. Yeah. Okay, it's okay. All right. Okay. Let's see. So this one here, basically the explanation is that R protein is required, okay, to have protection against S. So that is kind of like redundant, but that's basically it. Without the R protein, the culture has difficulty growing. So something like that. Okay. So R, so what does it explain the effect of expressing S1? Yeah, without R protein, what you should have written is that without R protein, there's no protection. So there's no protection against S1 nuclease. Therefore, so now you can kind of tie it together, right? Therefore, cell density decrease from 7.3 to 4.5. Okay. And there it is. So you don't need the top portion, yeah? Okay, so this would be just 1 point. Let's look at the second part of this question. Describe the effect of expressing combinations of S and R on the survival of culture 2 and culture 3. So you got to make reference to culture 2 and culture 3 and ask and make sure you answer the question. So I'm going to start by saying that, okay, in culture 2, okay, so what am I explaining? The effect of expressing both. Okay, R1 effectively produce, right? R1 effectively protected, right? The bacteria, I'm just going to say the Bt bacteria, okay, against S1 protein. Okay, so describe the effect, right? Nuclease activity. Okay, so you can even quote that this protection is 100%. Okay, in culture 2, okay, in culture, what is it? In culture 3, okay, R2 did not protect effectively, right? Did not protect Bt bacteria against S1 effectively. You don't have to use friction, quote data, or so this would be perfectly fine. Okay, so can you give yourself, this would be a 3 points question, 1 point for explanation and 1 point for each simple description for culture 2 and culture 3. Okay, so that would be it. So this would be 3 points for the first question, 3 points for the second question. Let's look at part C. Okay, so any questions so far? If you have a question, you can either type on the chat, I usually catch them, or you can just speak out of the microphone, okay? All right, so part C says, in naturally occurring solid environment, let me look at this one. In naturally occurring solid surface environment, such as soil, bacteria use the same signaling pathway to initiate formation of biofilm. So what a biofilm is described for you, yeah? So biofilms are densely populated aggregates of bacteria. So an example of biofilm is like your plaque, yeah, on your teeth. So there's a film of bacteria. That's why you want to floss and brush it every day. When that plaque can build up, okay, and the bacteria can cause tooth cavity as they penetrate the enamel, okay, into the dentine, right? So in the center of the biofilm, cells are more likely to interact with only cells of the same bacterial strain. Okay, so we're told that we can apply the same concept, same signaling pathway, contact-dependent inhibition, releasing a protein, and that kills the other cell. So they also told you that in the center, you only have the same strain. At edges, you are encountering different bacterial strain, right, at the edge. So there's a difference between center and edge. Identify the most likely type of ecological relationship among cells in the center of the biofilm and the edge of the biofilm, right? So center and edge in this case. Well, provide reasoning support researchers' claim that bacteria cannot form biofilm at low bacterial density. So how do you answer part C, the first part? Okay, we have to talk about identify the type of ecological relationship. Well, so what kind of ecological relationship do we have? Well, if you didn't know or can remember them, they actually reminded you in the first sentence. You can cooperate, you can compete, right? You can respond and sense to other strands. So it will be those two terms, okay? And you can also add predation, right? They can prey on each other. So is it going to be cooperate or compete? So the most likely ecological stream at the center, okay? So because of the same strands of bacteria at the center, so we're going to argue that they are not going to kill each other, yeah? Okay, so they probably will cooperate, right? Help each other because they're the same strand, okay? So it asks you to, what? Identify, right? So do you need to write a bunch of explanations on why you believe that there's a cooperation? No, okay? So in the center, okay, but the explanation is probably because they belong to the same strand, okay? Same bacterial strand, they want to help each other to form a colony of bacteria. So in the center will be cooperation, okay? And at the edge, well, at the edges, you're encountering other strands, so the most likely would be competition, yeah? With other strands, okay? All right, so that would be that part, just to identify. You know, you don't have to explain anything. Now, the second part you do, provide a reasoning for the claim that bacteria cannot form biofilm at low density. So the reason is basically at low density, there are not enough bacteria to actually to be close enough contact to trigger the contact dependent communication. So the key here then is from the original sentence here, the contact dependent communication. So you can see that when it's crowded, yeah, they're in physical contact, they're able to send proteins to the surfaces and the other guy can internalize it. But if they're far apart due to the low density, such in communication is not physically possible. So you cannot form biofilm because basically that's what I said, right? So now we have to type it out again. Okay. Low density or low bacterial density. Okay. So there are not enough bacteria, okay, in direct physical contact to initiate contact dependent communication, okay, to form biofilm. So like that. So they didn't really explore a lot of what's going on with the data. So there's graphing here and a simple interpretation of the data and then one application. So that's usually the format. The last one is usually a little bit harder and they want you to kind of tie things together, apply to something else with similar mechanism, okay? So this one will be so this will be a 3-point right here. So again, why is a 3-point? You have, again, in the center, cooperation is 1 point, competition another point, and explanation, your reasoning is another point. So this is actually a 9-point question, yeah? So, but I want to make it 10, so it's a little bit easier. So it's kind of range a little bit. That's why I offer sometimes it's kind of confusing. What is a total point? Because it really depends on the test. So, okay, so let's give this, okay. Let's make this 4 points then, yeah? So this would be 1 point here, 1 point here, and the last one, if you answer correctly, okay? So you have to mention the contact inhibition, okay? And the fact that there's not enough bacteria, and then you have to be in physical contact, right? And contact-dependent communication. You need that word in there. That's the whole point of this part C. So there's 2 points, and 2 points, so 4 points for part C, and then 3 points for part B, and then 3 points for part A. So can you give yourself out of 10, please? So we need to collect the score from you. Okay, that's not, we're gonna do that every week. So make sure you try to get the test done. Now, this is what the AP booklet kind of look like if you haven't seen it before, right? Okay, all right, let's do the next question. Let's move on. So the next question here it is, okay? So we given this data. So usually I like to look at the data and what does mean. Yes, but this one is kind of difficult because you have the y-axis called aggression score, and the x-axis are a whole bunch of letters. And you got to read. Okay, what is M? Similar to males living in Makara Island, it just doesn't make sense right now, right? So then don't try to spend too much time on this. But the figure one kind of explain it, right? The behavior of male flycatcher, that's kind of some kind of birds on Makara Island to different combination of bird, model, and songs. The condition for each treatment are indicated as ±2 standard error of the mean. That's SEX. That would be the standard error of the mean. The X is the mean. Okay. All right. So here's the question. In an investigation about the roles of visual and auditory cues in mating competition. So that's the title. That is the subject here. We want to find out the role of visual, so that means looking, right, at a particular bird, and hearing, right? So visual would be what they look like, what the birds look like. Is there any visual cues, colors? And auditory cues would be the chirping, right? The mating calls, for example, in mate competition. So it's competition for mating. Researchers studied two distinct populations of flycatchers. Okay, so we have flycatchers are birds, a species of bird that lives in the Solomon Islands. So they tell you that we got these birds, and there's two different populations. It's one species though, because it says here is a species of bird that live on the Solomon Island. There's just two separate populations, but they're the same species. And that's why they compete for mating, right? Ability to interbreeding. Okay, and then the question is, flycatchers on Makara Island have light brown bellies, and those on Santa Ana Islands have uniform black. So that's important to note, right? So, and then we have the M Island and SA Island. So M is light brown belly, and the other ones are black. So that would be the visual, okay? All right. The songs produced by flycatcher in each populations are also different. To investigate the songs are different, right? So they sing the songs. To investigate male flycatcher competition, again, that's what the whole point of this is, mate competition. Researchers exposed male flycatchers from the Makara Island, right, that's where they're found, okay, both of these are found, to different combinations of, number one, bird model resemble male from each of the two populations. So that would be visual. So you present them with birds that looks like male from each of the two populations. And two, recording of the song. So that would be auditory cues, right? And see how they respond to it. Okay. And then number three, models and song recording of a different bird species, the golden whistlers. So the different bird species is like a control. So golden whistler is a different species where the Makara Island flycatcher and Santa Ana Island flycatchers are the same species, okay? All right. On Makara Island, the researcher located territory defended by single mating pairs of flycatchers and assigned each territory to 5 different groups. So now let's see if these groups makes a little bit more sense. Okay, right here. So I'm gonna actually make a copy of this, and then so that we can get a better visual. So now it kind of makes sense why the y-axis is aggression score, right? So the behavior we're trying to monitor is the mating behavior, and the way to monitor that is by looking at are they aggressive or not. You would imagine that if there's a fierce competition for mating, the mean aggression score would be high. So you're aggressively to each other, right? So if you're presented with visual clues, are they aggressive? If they hear the songs from different groups of birds, are they aggressive, right? Okay, so if you look at the data again, so I have the data now on the left side, okay? All right, so for each trial, the researchers observed the behavior response of the male flycatcher and assigned aggression score from 0 to 10, okay? Mine is partially cut off. Okay. And then the question is, a higher aggression score indicated more aggressive behavior. Sure. So let's just try to look at the data and see if that makes sense to ourselves first before we answer the question, right? So you have 5 treatment groups. You can see treatment group 1, 2, 3, 4, 5. the 5 different groups. So what does it mean? For example, if you look at treatment group number 1, okay, what are we doing? So we have M and M. So what does that mean? If you look at the figure 1, it explains one more time. The behavior response of a male flycatcher on Makara Island. So to different combination of bird, models, and songs. So this means that you're actually presenting to the Makara Island flycatcher. Remember, if you look at the beginning, they are the same species. There are 2 distinct populations of the same species. One group live on the Makara Island, the other group live on the Santa Ana Island. So you're presenting to the Makara Island flycatchers. So you're presenting them with what? You're presenting them with treatment group number 1. You're presenting these birds with Makara Island and also the songs from Makara Island. If you look at the treatment group 2, you're presenting to these Makara Island birds visually with Makara Island birds, but song-wise, you present to these Makara Island flycatchers with the songs from the Santa Ana Islands. So the treatment group is on Makara M birds, okay? Birds from M, not from SA, from M. So you present to M, okay? The birds on the M group, okay? So if you look at this one, seems like they're aggressive. If you present them with M, so they compete with each other for mates. So because they're same species, they look exactly the same. So 1 has a very high aggression score. Now, if you look at treatment 2, kind of similar, M, right? Also trigger a lot of aggression, right? The songs are from somebody else, so I guess that is a little bit better than if they were both cues, right? Visual and auditory, if they're both from Makara Island, then you get the most aggression, okay? But if you look at the group 3, there's a significant drop. So look at 3 and 1, those will be good comparison. So 3 and 1 tells you how significant the visual is the appearance is, right? The visual model, if you change to SA, aggression dropped tremendously, yeah, when you don't change the song. So it allows you visual. And then you have 3 and 4 that. So even if you change the song back to SA, sorry, I might be lagging. It says internet connection is unstable. So the songs, if you change to SA, it doesn't really affect the aggression. It's the visual that play a huge role. Okay, if you look at 1 and 3, right, visual plays a huge role in aggression, more so than the song played in triggering aggression from these M birds. Okay, and of course the golden whisper or whisker, whatever. So G, this is like the control for everything. So if you present the M birds with another species, golden whiskers, visual and song model, they don't have much aggression. There's no reason to be aggressive to them since these ones do not compete for mating. So group 5 must be the control group, okay? All right. So guys, can you guys look at the question? Yeah. And if you haven't done it, can you try it? Part A. Let me go grab something. I'll be right back. Can you give me a sec? Yeah. You guys are trying, right? Okay. All right, thanks for waiting. Let's take a look at this. Okay, so let's look at part A. Yeah. Based on information, identify one independent variable, that would be the x-axis, and one dependent, that would be y, because y depends on x, right? And one negative control. The negative control will have to be group 5, because group 5 is a completely different species, golden whiskers. They are not going to compete for mating. So that will be a negative control. Positive control will trigger the behavior, which will be 1. So group 1 will be the positive control, because you're presenting the M birds with males from M and songs same by M, right? So 1 is the positive control, and 5 will be the negative control. Yeah. Okay, does that make sense? Yeah. So let me just write that down. Okay. So that will be 1 to 3 points. So A. Let's write down. Yikes. So much lagging. Okay, so A. So the independent variable. So this would be the. So what would be this? The treatment groups. Okay. So treatment groups. With different models, right? With different. We can just say treatment group, yeah. Models, visual and auditory models, okay, with different visual and sound model combinations, right? And dependent variable will be the mean aggression score, right? So try to use the same word. I think they say mean aggression score. Mean aggression scores. And again, this is about mating, which is very important. In terms of evolution, mating is the only reason for existence. It's to survive, reproduce, and pass on characteristics. So that would be the only reason for existence. And the negative control. So the negative control we talked about is the one that will not trigger your response, which is aggression. So negative control must be 5, and then positive control is not asked for, but I just want to put it here. The positive control will be 1, okay? Because positive control will generate the aggression, and that will be the males that look like the M birds, and also the song sent by the M birds. All right, so that's a very easy 3-point question right there. So you can see it's not all about writing essays. Okay, so usually they start with those. So this will be the experiment one, yeah? We did the graph one, this is the experiment one. Okay, let's do the next one. Based on the data, make a claim. So that's also a type of question. You gotta make a claim. It's like write a hypothesis or write what you think about, right? What about the behavior response of the male macaira flycatchers to a model of another macaira flycatcher? We know that triggers aggression. So that will be the claim. You can't go counter the other way, which will counter the data given. So again, the claim is, what would be the behavior response of the M birds to another M bird? And right here tells you, right here, right? You present the treatment group is this. This is group 1. When you put M birds, it's lots of aggression, but you need something to compare with. So I'm going to compare with group 3. Because group 3, treatment group 3 and 1, the difference is the visual, right? The M birds versus the SA bird. Okay, you can even bring 5 in, or no 5. Yeah, sorry. So it will be 1 and 3. Okay, so those are the one I want to focus on, and use the data to justify the claim. That's why we were looking at the data, right? Okay, so that would be that, right? Okay, so and then use the and then okay, so that would be that one first. So this is the part B. So now, given that, we're gonna be able to put into writing. Okay, so part B. Now, there's also other possibilities that you can write about. Okay, But so this is just one possible comparison. So I'm actually going to do that first. Okay, so let's look at this. Yeah, so we're asked to talk about my claim. Right, so here's a claim that birds, male birds from Macau Island show lots of aggression. Okay, lots of aggression towards other male birds from Macau Island. Yeah, so I know it's kind of like self-explanatory, but that's what this kind of test is. Yeah, from Macau Island. And use the data to justify. So this is my justification. Yeah. Now you don't have to really write it out that you're doing justification, doing claim. It's just easier for us to do that at this point. So how do you justify it? Yeah. Comparing groups 1 and 3. Varies only in visual response. We see that aggression, because you got to use the data. So I would point out number every time they ask for data. I would point out numbers, right? If there's number possible, we see that aggression. Okay. Trying to wait for me to go in here. Oh, good Lord. So this lagging is just not very good. Okay, so let's see that. Okay, so we see that aggression is significantly higher in 1. So this one, I'm gonna remind them is that this one is the with M birds and a lot lower with in 3. And 3 again to remind them this would be with the SA birds, right? So that would be Santa Ana birds. So this means that it's actually quite aggressive, right? Okay, so again, I'm going to look at what the question is asking again to see if we fully answer the question. Make claim about a behavioral response to the visual model of another M flycatcher. Okay, so we don't have to compare to the song, because I was initially thinking, okay, do I need to compare the difference between the responses to a visual versus the auditory? I don't have to do that based on the question, yeah? And then the second part, make another claim about the behavioral response of male M birds to the songs recording by another M male birds. Use the data. So I kind of do the same, but just now with the different groups, right? So which groups should we focus on here, right? Since we're doing the songs, there's actually 2 possibilities. You can look at group 3 and 4. So again, there's different answer now here. So I can't really tell you that you have to do a certain way. If you look at 3 and 4, that would be keeping the visual constant. And once you change the recording song, the aggression doesn't really change very much, right? So you can use 3 and 4. You can also use 1 and 2, right? Keeping the visual the same, and if you change the song, the aggression dropped a little, right? So either way, comparatively, they're just not as aggressive to songs as they are to visual, yeah? Okay, so my second justification, my claim, is that what we're doing? So make a claim about behavior to the recording, yeah, the Makara song. So now I actually want to talk about compared to group 5. I think because we haven't been using the control, so I don't feel good about it. So again, if you look at this, make a claim about the behavior. So I'm looking at the second part. Well, the first part doesn't matter. Do I need to talk about the control, the negative control, right? So make a claim about the behavior response of to the song, okay? And use the data to justify. Yeah, so to be safe, it's best to bring in the negative control. So if I go back to the previous question, the claim, and how do I introduce the negative control in here, right? So my claim is that male birds from this one shows a lot of aggression towards other, okay? Yeah, so I'm gonna put in justification here, okay? So this is 1 and 3. In both groups, aggression is a lot greater than in both groups. Aggression is high relative to control 5. Okay, so I already talked about what control 5 is the negative control. So I feel better if I include that. Okay, second one is the claim about the song, right? So I'm gonna make a claim, and there's no right or wrong. So I'm looking at groups 3 and 4. So that the male birds. So learn how to write these because it's kind of weird when you trying to put them in words. Okay, it feels different. Okay, so male birds from M Island show little aggression. Okay, little aggression. I don't have to compare with anything. This is my claim. Little aggression to other towards other to the songs. Sang by shows little aggression. Or maybe I should say that my claim, no, there's no difference. Aggression, little aggression, okay, and so make a claim about the behavior response of this to the songs, right? So they're not very, they don't show aggression. So male birds from M Island show little or no aggression towards the song sent by other M birds, Makara birds, yeah? So again, that would be 3 and 4. Okay, if you look in 3 and 4 again, okay, 3 and 4, the difference is the songs, right? And whether you present them with the same species M song, with the M song or with the SA song, they're the same species again, okay? There's little aggression difference, right? So my justification, okay? Looking at groups 3 and 4, the aggression score is similar whether oops, sorry, not this weather. We're gonna get rain this weekend. It's similar whether and male, whether the M birds presented with songs from M birds or from SA birds. So I'm trying to say that whether you present to these male Makara birds with the songs other male Makara birds sings or songs sent by other SA male birds, there is no difference in aggression. Yeah, and there's some typos. I just don't want to go back and fix them very much, so don't worry about that. Okay. All right. So I feel that I don't have to compare this to the control. There's no point of using the control here. Okay. Well, I could. Yeah, I could. Yeah. Okay. I could talk about the control. So maybe because again, the control is that in either case they're, even though they're higher, but I don't think that's the point here. Okay, so you don't have to if you don't want to. Okay, so that would be it. So this one, if you look at the point system, then identify. Well, that would be 3 points. I think we talked about that. And this one, make a claim 1 point, justify 1 point. Make a claim 1 point, justify. So that's 4. 3 and 4 is 7. So do you see part C? You have 3 points left. That's why there's identify 2 and identify 1. So there's 3 more points, right? So most of the time, you can kind of gauge, well, what are the points? Okay, so let's look at part C. So researcher claimed that the M island and the center island flycatchers are diverging into different species. So that would be divergent evolution. Right now, what's going on is that they're the same species, and we kind of it's kind of told to us right here is a species of birds, but they live in, they're 2 different populations, 2 distinct populations of the same species. Okay, so it's the same species, they're not different species. So, but they look different, right? The SA is black, and the other ones are light brown, and the songs are different also. So, seems like they're the same species living in 2 different populations. They're about to diverge. So, divergent evolution will be on the way, right? So they live in different environment, they have different physical behaviors, right? So that's probably causing them to diverge. So that would be like that, okay? So divergent evolution. Don't forget, divergent evolution produces homologous structure, okay? So homologous structures, like our arms and bats' wings, they live in different environment, but they have similar ancestry. Well, that's not what the question is asking. It's asking to identify 2 potential pre-zygotic barrier that is causing the divergence, that is promoting the speciation, right? That is preventing interbreeding. So the pre-zygotic barrier here, we have, we cannot just talk about any one of them. So the pre-zygotic barrier to interbreeding, so these barrier to interbreeding keeps the population reproductively isolated. And if you isolate them long enough reproductively, then they eventually will become different species, okay? So there's a habitat, okay? So let's just review these one quickly. There's temporal isolation, timing, like breeding timing, seasonal breeding, right? There's also mechanical, okay? Isolation. There's also ecological niche will be different, and there's also genetic isolation, right? So you can't just pick a random 2, because, for example, I cannot pick mechanical because I don't know the mechanical anatomical differences between the birds. Do they have different reproductive anatomy? We don't know that. And do we know anything about the genetic? Probably not. Do the gametes fail to recognize each other? That would not be the best answer for what's contributing the divergence, right? So seems like it's the sound and visual. So you can argue 2 of them, right? Pre-zygotic barrier that may contribute, and then identify 1 piece of evidence that indicate that speciation already occurred. Okay, so let's answer part C here. So what do you guys think we should put here for part C? Okay, so part C, and actually what did you pick, right? What do you guys put here? So again, I need to click on this, and then to zoom it, it takes time, and I'm gonna stretch it out, so, and then I'm gonna move it, stretch out a little bit more. Okay, so the 2 pre-zygotic barrier looks like they have different songs. They don't live in each other, so I'm going to say habitat. Now, do you have to explain your choice if the question says identify 2 potential pre-zygotic barrier that may be contributing to the divergence? So no, you just put down 2, most likely one. You don't have to explain it. So because they live in the Solomon Island, but 2 separate populations. So I'm going to say it's maybe ecological and behavioral isolation. Okay, hold on. We have temporal, mechanical, behavioral isolation, not ecological. I don't know what I was thinking. This is a behavioral isolation, yeah? So number 4 should be behavioral isolation. So if you can't do the dance, sing the song, you cannot attract for mating, yeah? Okay, so this would be definitely one of them. And the other one I'm gonna put down is maybe habitat isolation, okay? Since I am not told their breeding seasons, I want to stay away from that. So I'm going to say ecological, not ecological, there you go again. Behavioral isolation. And the second one will be habitat isolation. Because although they're found on Solomon Island, they're found as 2 separate species. If they're separate species, separate populations. So that means they're not in the neighboring potentially, yeah, necessarily. So maybe habitat, okay? And you can put anything else because there's not enough clue. So if you put like behavioral, if you put temporal, that's fine too. So there's no, you can even put genetic and mechanical because there's no real concrete evidence that says otherwise they're not mechanical, they're not genetic. So you can pick any 2, okay? So is that okay, guys? Any 2 from the list, okay? All right, second part. Now you have one piece of evidence that speciation already occurred. So what is the evidence for species? Yeah, ability to interbreed, DNA evidence, right? So they look at ecological niche. Are they the same or different, right? So if they have very different ecological niche, that means the speciation has already happened, right? If they lost the ability to interbreed, so that would be one as well. Okay, so lost the ability to interbreed, yeah. The M and SA birds have lost the ability to interbreed and have fertile offspring. Yeah, so always put that in. So because if the offspring is not fertile, like mules, right, when donkey and horse mate, that's not a that is a barrier as well. So if they lost the ability to breed and have fertile offspring, indicates speciation already occurred. Okay, all right. So if you tweak this and talk about the DNA evidence that they're different enough, that would be fine too. So there's no right or wrong answer there. Okay, so can you give yourself 10 points, please? Again, the first one is 3 points, part A. Part B is 4 points. And part C is 3 points. Yeah, so please add it up. These are 10-point questions. So those are the 2 most engaging ones. We're done. So let's move on to 4-point questions. So 4-point questions are very, the point system is really easy. So let's take a look at this, yeah? So the 4-point question here. Still a little bit lagging. I'm gonna see if I can zoom in this one. All right, let's look at this, yeah? So we're looking at the bees. Now, in case you didn't know, bees and moths, they have very long tongues, and there are special adaptations in order to reach to the base of the flower for nectar and whatever nutrients they're looking for. Okay, so this is, and then you would, when they do that, they bring pollens from flower to flower. That's called cross-pollination, right? Okay. So this one seems like it's looking at that. Okay, so the question says, bees use the tongues to access nectar as food source. Oh, that's good to know, so already know that. From the base of the flower. Flower vary in depth from shallow to deep. Okay, so some flowers are very shallow, so bees do not have to use long tongues, but they can reach, and some are very deep, so you have to be very specialized. And that's what co-evolution is. So the bees or the moths will co-evolve with those flowers. So the deeper the flower is, the more reciprocal adaptation or co-evolution has happened for bees with long tongues to specialize on them. So no other bees can do that unless they have the same long tongues. So that would be a co-evolution reciprocal adaptation, okay? That's another way to kind of think about coevolution. So flower varies in depth. Okay, good. And then, so let me take out this again. All right. And then, so the tongue is short to long. So we had shallow and deep, short to long. So use the same terms. So don't refer to flower using short when they use shallow, right? And don't, so that's what you have to say. All right. In many species of plants, depends on bees to transfer pollen from one flower to another for fertilization. So that's the benefit of bee. This is mutualism. Bees transfer flower for pollination, so plants get what? What is the benefit of cross-pollination? The benefit of cross-pollination is genetic variation, genetic diversity, which will equip the flowers for potential environmental changes, right? You always want to have genetic variation. And for the bees, it will be food. So mutualism. Researchers conducted a graph to illustrate the conditions under which short tongues provide bees with advantage in an environment, okay, where short tongue and long tongue species are present. So we have a graph. So we're going to go back to the graph. And what does the graph illustrate? The conditions under which having short tongue provide bees with advantage. So what is the advantage of being short tongue bees, right? Okay. And the environment has both kind of bees, okay? Short tongue and long tongue bees. Okay, let's look at figure 2 and see what that means. Okay, so we're going to look at figure 1 and 2. So figure 2, relative advantage of short tongue bees. Okay, that will be the y-axis. Okay, let's take a look at that. So this is the y-axis right here, the relative advantage. So when you look at the relative advantage, 1 means there is no advantage. This would be the same as the control. If you are greater than 1, that means that you're more advantageous, being okay. And then if you're less than 1, you're less advantageous. So this is relative to the short tongue bees. Okay. So relative advantage of short tongue bees in relation to flower density, which is on this axis right here, Z. Okay, you see the density increases 2 to 10. Okay, so do you see that? Based on that, just look at these two matrix, Y and Z. Yeah, if you are short tongue, right? When the flower density is high or low, it changes very little. You see that? It doesn't change very much. So your relative advantage remained about 0.7. Whether you have 10 flower density, 10 flower per meter square, flower per meter. There's not much. Maybe there's a peak here. Isn't much. And then the other axis, there's gray shading, huh? Well, that is not good. I don't know which part is shaded gray, I can guess, yeah? So the other axis will be the proportion of deep flowers. So do you see that when the deep flowers are very deep, right? 1, that means a lot of the flower, 1 means 100% of the flowers are deep. Then the short tongue have very low advantage, so less than 1. But when there's very few, maybe 20% flowers are deep, the rest of them are shallow, then they actually have an advantage. You see that being short tongue has advantage, but most of the flowers are shallow. That's why 0.2. 0.2 means more shallow. 0.2 deep means 0.8 shallow. So that's what this axis is saying. So as the flowers become more shallow, the relative advantage quickly increases. Yeah. Okay. All right. So, and the shaded region, I don't know where it is. So I think it's kind of like shaded. I see little dots here, but sorry, this is should have caught this. So I'm sorry. This is one I don't have the file. So I think let's answer them, okay, and see what happens. Bees using their tongue to access nectar. Actually, we're ready to answer the question, but let me take a picture of this. So I have a feeling we need to review. Look at this again. Let me just take both. Yeah. Alrighty. So this will be the picture that we need. Okay. And let's move this away. Let's take away this. Let's move this back. And we don't want this. I don't need that. Okay. So let's look at this question. Yeah. So sometimes they give you difficult graphs, and now do you understand how to read a graph with 3 axes. You just compare them all to y, but the flower density versus relative advantage, and then the other one is the proportion of deep flowers. Okay, one means 100% deep, and you can compare also that to. Okay, I'm trying to move this thing. Oh, great. Okay. Okay, let me move it again. Alrighty, so let's do that. Okay, so the first question here is talking about identify the environmental conditions. So they tell you what those conditions should be. So what are the conditions? Flower density and what percent is deep flowers, where short-tongue bee has the greatest relative advantage over the long-tongue bees, right? So what do you think? So you want the short tongue to be more advantageous relatively, yeah? So that would be greater than 1. That would be greater than 1, right? Okay. So in terms of the flower density and proportion of bee flowers, so you want more shallow flowers. Again, it's hard for me to guess where is shaded. So I'm going to try to shade it right now, yeah? So it looks like all these are shaded here. Okay, all these are shaded. Let me use a little bit thicker. So all these are shaded, shaded. Look like all these are shaded here. So these are all shaded. Okay, I see the little dots on them. Okay. So these are all shaded here. Based on what we shaded here in pink, we can use this data to answer the question. Yeah. So maybe like here and here, these are shaded too. Okay. Maybe like that. Okay. I think that's kind of, yeah, maybe those two are not shaded. Are they? Ayo. Yeah, it's hard to tell if those are shaded or not. But anyways, maybe we don't need to be that precise. Okay. So again, the question is asking you to identify the conditions. So low or lots of deep or little deep, right? Okay, high density or low density. Okay, short tongue has advantage. So we're going to compare that to 1. Okay, so it looks like there's a little bit uptrend for the flower density. Earlier I said it's about the same, but since they asked you to compare, so there's a yeah, I see definitely a low proportion of deep flowers, and the flower density doesn't really affect all that much. So this one here is 1, I think it's about here. So this is where 1 is. Yeah, so I'm gonna say that because that's still below 1. So I'm going to say that the short-tongued bees are disadvantaged at all flower density, but it's more advantageous when the proportion of deep flowers are less than like 0.6. Yeah. Okay. Do we need to quote? Maybe not. I just said the environmental conditions, but it's hard to describe the proportion without a number. So do we need to quote a number? I would do that for this case, right? So short tongue bees are bees at a disadvantage for all flower densities. Okay, because even the highest peak towards the 0, it is still less than 1. So that means you're not advantageous, right? The short tongue ones are not relatively, right? All flower densities and are advantageous when portions of deep flowers on the test. Try not to abbreviate. R is less than what? What do you think here? So it looks like the shaded part there is less than 0.5 or 0.6. You start to peak up, right? So I would say maybe 0.5 or 0.6. Yeah. Okay. Less than 0.5. Okay. There. All right, so that will be the identification. You don't have to justify it. Again, identify or to state something, you usually don't have to justify. Okay, let's look at the next one. We're not done with this because they also ask you to identify the range of proportion of D flower at which long-tongued bees are always has advantage over the short. Okay, it's kind of redundant because, okay, so let's look at that, right? So we also have to identify that. So let's come back to my graph again. You want long tongue to be advantageous, so that means the short will be not. So you want the bottom part, okay? So the long tongue will be advantageous. Looks like it's the shaded part here, right? So that looks like 0.7 or higher. Okay, so 0.7 or higher proportions of flowers are bees. So long tongue bees are always advantageous. So you want to be higher than 1, okay? So it's really hard to read this, but I would say 0.7 or higher or 0.8. You want to be always, right? And when the proportion of deep flowers is 0.8 or higher, okay? That 0.8 is for sure, yeah, and 0.7 maybe. So they'll give you a leeway on the answer, okay? So can you give yourself 2 points here? One is 1, 2, 3, 4. So one identify, you got to identify both. If one of them is wrong, then it's wrong in this case because there's only 1 point assigned. So you got to talk about both environmental conditions and get both correctly to get that 1 point for identification. Same thing with and then the second identification is this. So 2 points, yeah? Can you give yourself out of 2 points, please? All right, let's look at the next one. Bees with short tongues were able to get nectar from deep flowers by drilling holes in the base of the flower without visiting the top. That's cheating because flowers, from the flower's point of view, you want the bees to go to the top where they can get cross-pollinated. They can get pollen from other flowers so they can get genetic variation. But these, they go to the base of the flower, they just make holes to get nectar. So these are cheaters, yeah? Okay, so that would be a disadvantage for flowers if they cheat because flower does not get pollination. That's what it wants. So it becomes a parasite, for example, like a pest, right? So the flower lose nectar, nutrients, and then they spend energy to make nectars. They get no cross-pollination. All right, so what is it saying here? Okay, so this behavior does not kill the flower. That's important, right? In particular environment, bees with short tongue replace bees with long tongue. Okay, in a particular environment, bees with short tongue replace bees with long tongues. Okay, so we have this environment that's all short tongues. Yeah, I think that's what they're trying to say here. So we have all short tongue bees in this environment. So they're all cheaters, right? Predict the effect on the proportion of plants with the deep flowers in future generations. Justify. So these are cheaters. So what do you guys think? This one here. These with short tongues are cheaters. It does not kill the flowers. So this means that the effect of proportion, whoops, let me look at this again. Effect of proportion of plants with deep flowers in the future generation. So the deep flower are no longer selected for, right? Before they have advantage, right? They get pollinated when there's long tongues around. So without the long tongues, the deep flowers are not pollinated, so they don't have a selective advantage, right? So I think because of that, the proportion of plant with deep flowers should decrease, right? Because they're no longer at an advantage. There's no long tongue bees to pollinate them, and so they're just equal, right? If you're a short tongue bee, you can visit a flower with long and flower with a deep a deep flower. If you are short-tongue bee, you can also visit a flower that has shallow flower. So it makes no difference. You see a long one, a deep one, you make holes, you get nectar. You see the short one, and you just pollinate regularly. So there's no difference. There's no selective advantage anymore. Okay, so the proportion of deep flowers should decrease. You can also say remain the same because there's no selective advantage. So you can argue either one. So even if you put down should be constant because there's no more selective advantage, that's fine. But you just have to justify your prediction. So sometimes these justification one, there's more than one possible answer as long as you can justify it. So I'm going to say the proportion of deep flowers should decrease as they no longer have any selective advantage. In fact, they actually have a disadvantage because they don't get pollination, right? When in an environment with only short-tongue bees, okay? Short-tongue bees. Without cross-pollination, these deep flowers may be are not as selective, yeah, not genetically diverse enough. I'm going to say that they're not genetic and are selected and may be selected against, yeah, because I'm arguing a go down. So deep flowers are not genetically diverse enough, and selected and maybe or are selected against. Because we don't know how long it will happen. So that would be my justification for it to go down. If you're saying that there's the deep flower proportion is constant, then you're saying that okay, there's no selective advantage for them, so there's no reason for them to increase. That would be fine. But if you argue that they will go down, then you have to kind of talk about they no longer have a selective advantage, or you can say that the shallow flowers are at an advantage now due to genetic variation. So either one. So their proportion will increase, hence mathematically will drop the proportion of the deep flower. So either way, okay, so you can argue either way. So no right or wrong answer. So make sure you get, can you score out of 4 points, please? Per payment, share, that would be awesome too. Okay, is that okay, guys? So far? I'm going to actually shrink this one, it's too big. Okay, all right, let's look at the next question. So please give yourself out of, what is 4 for this one, please? Yeah. Okay, I'm going to shrink this and then make this bigger here. All right, let's look at question number 4. Okay, oh, so here it is. So this one, okay, we have DNA, mRNA, and protein. So gene expression, yeah. So the DNA, we have 1, 2, 3, 4. We called exons. Those are coding, and the lines in between them are introns, and you can see they were cut out when you have the mRNA. So I'm looking at here the 2 strands of mRNA, 1, 2, 4, 5, 1, 3, 4, 5. This is called alternative splicing, right? Alternative splicing. So we actually get different exons are pieced together here. Okay, all right. And then when they're translated into proteins, and seems like the color coded for us. Okay, so guys, I'm going to ask you some questions. Can you look at this before we start so we can generate some questions, make sure we understand what we are looking at. So I'm going to try to make up some question here. So question number 1. So this is 1, 2, 3, 4. So question number 4. Okay, I'm just going to make up some question here. Yeah, number 1. Okay, so now remember when you talk about a membrane protein, there's 3 regions you can talk about. Okay, So I'll do the questioning a little bit. I forgot I want to talk about this. So there's three regions of a membrane. There's the transmembrane region, which is the region that is within the membrane. So they use the word transmembrane. Yeah. So the transmembrane domain or transmembrane region, that is always the part within the membrane. Okay. This is the cytoplasmic domain. So cytoplasmic domain is the part of the membrane, the part of the protein that faces the cytoplasm, and this is the extracellular domain. So the extracellular domain is the part that faces the what is that? The outside of the cell, right? So you can see that if you have a protein, okay, so this is called an integral protein that cuts through the membrane, or transmembrane protein. So when you look at a protein like that, those are the regions, okay, and those are the terms: transmembrane protein, a protein that cuts through the membrane. There are three domains. Now, your first assumption is that the transmembrane regions are within the phospholipid bilayer, so they must be nonpolar. That makes sense, right? The transmembrane region must be nonpolar. That's where the phospholipid tails are located, and the extracellular must be polar, and the cytoplasmic must be polar. Cytoplasm has lots of water, extracellular has lots of water. So the outside portions are polar, the internal regions, the transmembrane regions must be non-polar. That's kind of assumed, okay? And looking at protein A, I'm looking at protein A and B. They have the same cytoplasmic domain. And can you tell me which exon is responsible for coding the cytoplasmic domain of protein A? Yeah, can you tell? Which exon number is responsible, right? So which exon is responsible for coding the cytoplasmic domain of the protein? And the answer would be number 5. So exon 5, right? So that's basically what the shaded regions are for. If you see the dark shaded exon number 5, it codes for that, okay? And then if you look at the transmembrane, what about transmembrane? Transmembrane region, 5, exon 1 and 4, right? Does that make sense? This should be pretty easy to tell using the colors, right? So what makes protein A and B different is the extracellular domain. Okay, so what makes protein A and B different? So the extracellular domain. Okay, so the extracellular domain is responsible for binding to ligands. Ligands are chemicals that is trying to communicate with the protein. And the intracellular domain is responsible for signal transduction. Remember, signal transduction has 3 stages. We have my protein is so bad, I'm just going to draw it again. Yeah. Okay. So we have the receptor binding. This is my phospholipid. You got a whole bunch of these, right? Remember the inverted phospholipid bilayer, right? So each one has 2 fatty acid tails. Those balloons with tails are phospholipid. So let's say this is my protein. Okay, and whether protein A or B doesn't really matter. So the point is that the intracellular, okay, extracellular. So intracellular is the cytoplasmic. So let's say we separate like this. Okay, like that. So this portion is receptor binding. So the extracellular domain is for step 1 of signal transduction. Extracellular domain is responsible for receptor binding. So in this case, protein A and B differs in the extracellular domain. So what does that mean? That means they bind to different ligands. So you can tell that ligand A bind to protein A, ligand B bind to protein B. They're responsive to different ligands. But if you look at the cytoplasmic domain, they're the same. If they have the same cytoplasmic domain, they probably use the same mechanisms, same enzymes, so they have similar pathway. So that, I think that's what these arrows are for when they converge to the same secondary messenger because of the same cytoplasmic domain. So that's likely that they use the same cascade of the reaction or the same secondary messenger to communicate. Okay, so we have that kind of assumption here, and those are reasonable assumptions that we have to be able to see. So receptor binding, and this is for signal transduction. Okay, the signal transduction via a secondary messenger and cascade of reactions. Okay. So this process of signal transduction is studied by molecular biologists very thoroughly. So this is actually a very important mechanism. Okay. All right. So alternative splicing is able to use the same DNA to make two similar but yet different proteins, protein A and B. They're similar because they have the same transmembrane cytoplasmic domain. They use the same cascade of reaction and secondary messenger to carry out the response, but they're different because the extracellular domain means that they can be responsive to two different chemical ligands. All right, let's look at this one. So cell signaling in eukaryotes is often dependent on receptor proteins on the membrane. Sure. During the production of mature mRNA molecule coding for these receptors, pre-mRNA are processed to remove introns and connect exons together. Alternative splicing, right? Explains why there's very little DNA variation, but there's tons of protein variation from the same DNA molecule. So it's very important to understand that. Okay, so different mature mRNA can be formed from the same pre-mRNA by alternative splicing, which results in different protein sequences in the resulting polypeptide. In this case, it's protein A and B. Figure 1 represents the expression of a gene with 5 exons, there it is, that can be alternatively spliced to produce a receptor protein A or a receptor protein B. Explain how A and B can cause identical cellular response in the cell. So how can they cause identical cellular response, right? Well, the answer is because they have the same intracellular or the cytoplasmic domain, right? Having the same cytoplasmic domain, okay, recruiting the same secondary messenger can possibly trigger the same cellular response. That's the answer. And that would be a good explanation. So maybe based on what we're told, right? Because nobody knows for sure. So based on the data and our understanding of the biological concept, that's what we would put as an explanation. Yeah. So both protein A and B, okay, identical cytoplasmic domain and recruits. And the second part is important, yeah, and recruits the same secondary messenger for signal transduction. Secondary messenger. Now, secondary messengers amplifies the original ligand A and B. Yeah, messenger, not massager. Messenger for signal transduction. Yeah. So this is how they would able to have the same cellular response. Okay. Part B. So okay. Predict the most likely effect of 2 nucleotide deletion in the intron. So no effect, since introns are removed and do not code. Since introns are removed and are not coding. So if you have a mutation in the introns, there is no effect on the structure of the resulting protein. Yeah. So your answer, you got 2 points here. Okay. So 2 points for part A, 1. Okay. 2 points for part A. How do you get the 2 points? You have to talk about the cytoplasmic domain. You can even say that they're both made from coded by exons 5 if you want, but that's enough, right? So this is the exon number 5, okay? Exon number 5, and also the same secondary messenger. So 2 points for part A and 2 points for part B. One says no effect, and then one for justification. Introns are removed. I'm going to say, instead of not coding, I'm going to say the remove and does not show up and not translated. Yeah, are not translated. That's a much better answer. Yeah, are not translated into protein. Okay. All right, let's move on. So give yourself out of 4 points for question number 4. We're going to move on a little faster. Number 5. Okay, we're talking about hormones. That's here. Now, leptin is actually a hormone produced by your fat cells. Okay, what does it do? Okay, it's pretty cool. It signals an organism to suppress appetite. So let's process that for a second. That means you're gonna feel full. Yeah, when you feel full, you suppress appetite, right? So you feel the fullness or satiation, right? So if you have leptin, you feel full. If you don't have leptin, you don't feel full, even though when you ate a bunch of food. Okay, all right. Leptin is released from fat cells in white fat tissues called adipose tissues. We have white fat. We also have brown fat. Brown fats are burned to produce heat when you're cold, and white fat is for storing energy. Okay, so what do? These leptin are hormones, so they always bind to receptors, right, on the cells in the hypothalamus. That's the brain region that controls your appetite. Okay, identify a way that leptin produced by fat cells in the adipose tissue in the abdomen, right? How can it send a signal to the endocrine cells in the hypothalamus in your brain? So how do you travel from fat in the abs all the way to the brain? You travel how? So you travel in the blood, right? All hormones are released into the blood and they travel to target organ. That's the answer. Yeah. Okay. So let's put that in. I'm just going to put that very quickly, and then we can move on. So this is question number 5. Okay. So let me move over. So this is number 5. So A, leptin is released into blood and travels via circulatory system or blood to hypothalamus. So hypothalamus, do you need to know what hypothalamus is? No, it tells you what hypothalamus is, right? That is in your temporal region, yeah, where the spinal cord ends up in your brain. All right, let's look at the second part. Researchers are investigating the effectiveness of various treatments of 3 individuals with a history of increased appetite. So these 3 patients, let's call them. Individual 1 does not produce leptin. Now, that's a problem. If you don't produce leptin, then you won't feel full. You need leptin to feel full, right? So that's why the individual 1 keeps eating. And he does have functional receptors in the hypothalamus. Individual 2 does produce leptin, but he can't feel full. Now have receptors. The receptor, even there is leptin around, you cannot receive the information and trigger fullness sensation, right? And individual 3 does not produce leptin, does not have functional receptor either. So do you see that individual 3 is the worst? First of all, you don't have leptin to control appetite, and you don't have the receptor to respond to leptin. That's horrible. So if I have to guess, individual 1, 2, and 3 would be the most overweight. Without leptin to feel full, he probably keeps eating. And even if he does have trace amount of leptin, he doesn't have the receptor, so he won't respond to leptin, so he'll keep eating. So probably 3 will be the heaviest out of the 3 individuals. Now, the first treatment involves injection of leptin into the blood, which will travel. The second involves gene therapy. So there's 2 treatments. One is injection of leptin, so give them leptin. Second, you got to have a gene therapy. What is the therapy for? That results in the production of functional leptin receptors. So if somebody does not have leptin receptors, I can give them a gene therapy, carry using a virus, inject into the bone marrow, and hopefully the cells will start to produce leptin receptors, so they will be responsive to leptin, right? So in the template, draw check marks. So that's what you have to do to indicate the individuals in which the treatment most likely results in appetite suppression, right? So which treatment works? and columns and row may have more than one check marks. Okay, so this is pretty easy. And that's it, huh? There's just the points here. So if you look at treatment 1, and of course you have to take this into account what the problems are, right? So here's your answer then. Individual 1 does not produce leptin and does have receptor. So if you give individual 1 leptin injection, he does not have leptin. That will help him. Yes. Okay. Now he already have functional receptors. So treatment 2 is not going to help him. I mean, not treatment 2. The leptin receptor gene therapy. Gene therapy will not help him, right? What about a combination of those two? If you give individual 1 the gene therapy and leptin, yes, because again, he needs leptin, right? So first and third column, first treatment and third treatment will help individual 1. Let's look at individual 2. He does have leptin, so giving him leptin is not going to help him. That's not the problem, right? The problem is that he does not have functional receptor. So individual 2 needs functional receptor. So you got to give him a gene therapy. So second column will work because that's gene therapy. The third column will work because you also have gene therapy. Now, individual 3 has neither. So he needs both present at the same time. So the last column. If you give him the gene therapy only, doesn't help him. He does not produce leptin for the receptors. If you give him leptin only, does not help him. He does not have the functional gene receptors, right? So you got to give them both. Okay, so that's your answer. Okay, so this will be this is 5, 6, 7, 8. Okay, so this is still a 4-point question. Yeah. So can you give yourself out of 4 points? Okay, so how do you get 4 points out of here? Because I don't have a scoring guide. I need a scoring guide for the points, but so you have 1 point, 2 point, 3 point for this. And if any one of them is wrong, okay, you don't get that point, yeah? Okay, so do you see that individual 2 has two check marks? Sorry, not 2 check marks, yeah? You have 1 more point from part A, right? So travel in the blood, okay? So that's 4 points here, okay? So 1 point for each row. They have to be exactly the same. If you have additional check mark or missing one, you don't get the point. And 1 more point for saying that leptin is released into the blood. That's how they can travel long distance from ab to the brain. Okay, so 4 points here, pretty easy. Let's move on to the next one. Alrighty. So next one looks like is evolution cladogram. Good. So that is good because these questions are usually not hard. Okay. So this is question number 6 now. Okay. So if you look at this one, we have 1, 2, 3, 4. So let's read this. We have this EPA. So this gene, okay, in human codes for transcriptional factor. Remember transcriptional factor? You need it to activate transcription to get gene expressed. Okay. So this is transcriptional factor in low oxygen conditions. So it will be triggered when there's low oxygen, such as those in high altitude. Okay, so people live in high altitude, like the Mongolians and so forth, the Highlanders, right? Okay, researchers collected DNA from several populations of modern humans, including Hans. So you have HYLTD. Don't try to pronounce them. Okay, so these are the modern human. They collected the DNA from fossils, blah, 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 right? Okay. The researchers sequenced the most common, this allele, okay, in the population and determined the specific pattern of variation called single nucleotide polymorphism. So don't get too bogged down on what that is. This is just variation, yeah? At 6 positions in each of the population in table 1. So here's the single nucleotide polymorphism position 1, 2, 3, 4, 5, 6. This is what makes them different. So how do I know who's the ancestral human? So they kind of tell you, right? They collected this one here, the D right here, the Denisovan, Denisovan. Okay, those are the most ancient one because that's a prehistorical population. That's the most ancient. Okay, so it says use the template, construct a cladogram based on the allele. So based on this, okay. So who's the most advanced and who's the most who's the outlier group, right? Circle the location where the most recent common ancestor of these positions are shown in the table. So let's see what we are given with. Yeah. Okay. So, oh yeah. So here, so they kind of already gave us a cladogram, so we just had to fill in. Okay. So let's take a look at that. So this is for question number 6. Okay. So you're basically looking at, instead of looking at characteristics like a cladogram, here, rather, you're looking at DNA differences. So the more different the DNA, the more distantly they are related. The closer the DNA, the fewer differences, the more similarly they're related, right? So it kind of makes sense. Okay, so this is the this is that. And we're told that this L is here. Okay. So so how do we look at this one, right? Most common reason ancestor. Okay. All right. So if you look at this, L, I'm gonna see who's the closest to L, and that's also closest to each other. So I'm looking at these two guys here. Yeah, this guy and this guy here. Yeah, who's the closest to L and closest to each other? So it can be L. So I'm gonna see compared to L, this is G A A A G A. So G A A A G A. So this is two differences, and this one is G A A G A A. That's one difference. All right. So these two are very similar to L. Okay. So there's two differences and one difference. And let me look at Tibetan. So there's 1, 2, 3. Yeah, that's way more different. Okay. So our conclusion is that the Huns, H and Y, are very different. Y is only 2 nucleotide different from L, and Huns, H is only 1 nucleotide different. So this would be you can put either one. Yeah. So this is like Huns, and this is the Yo. Yeah. Okay. Y. And then the Tibetan and the other one must be the other two. Okay, it doesn't matter where you put it, no, because that's how this kind of works. You can flip them if you want. All right, so that's it. Construct cladogram. We did circle the location on the cladogram that represents the most common ancestor of all the population here. Well, that's easy. It will be this one, right? The most recent common ancestor is right here. This is the common ancestor. Okay. So let me get rid of this so I can move on to the next question. Where's the annotate here? I need to get rid of this. Okay, so yikes. Yeah, I forgot that I wasn't on this. So again, this would be maybe H and Y, and this is the T and B. And this is the most common ancestor right here. Okay. So that is 2 points there. Construct 1 point. If you miss any one of those, again, you can flip-flop, but if you miss any one of them, no point. Circle that will be, and it says circle. I shouldn't highlight it, okay? All right, puppy. The gene. This is the gene that they compare. Okay, at 6 locations of the gene, okay? So the gene shows strong indication for positive selection in Tibetan. And what is the gene for? It tells you what the gene codes for. It codes for a transcriptional factor that is activated low oxygen, high altitude. So it is a positive selection. That means it's selected for, whoever has it are selected for in the Tibetans. Okay, a population located in the mountainous region. Describe how the specific gene pattern above become common in Tibetan population. Yeah. Okay. So the question is, puppy. It tells you that, so you basically regurgitate what we are told. We know there's a positive selection, but we got to describe. What are we describing? How the gene how the gene pattern shown above become common. That would be natural selection question, so you had to talk about it. But in an environment with low oxygen, right? As in the Tibetan, in an environment with low oxygen. So let me stretch this out a little bit. Yeah. The gene selected for with low O2. Okay. Yikes. O2, low oxygen. okay, which is high altitude. Yeah, this the EPAS1 gene is activated and expressed more frequently. Is activated and expressed. Okay, and expressed lagging. Okay, so let me see if I need to. Okay, Tibetan and denomination is 1. Okay, in this one, describe how the specific gene in the pattern, yeah, is selected for and became common. Okay, it must have an advantage. I'm trying to find out if there's any selective advantage. It has to. That's why they persisted in the population, right? Okay. The gene, the EPAS1 gene, must have survival advantage and is selected by natural selection. Okay. I would say that and individuals with individuals with the gene selected for by natural selection, survive, reproduce. So you regurgitate the concept of natural selection: survive, reproduced, and passed the gene there to the descendant, right? Reproduced and passed genes to their offspring. So that's why they become more common. So I think this question wants you to regurgitate the concept of natural selection. So upon reading it further, we don't know why having the gene is advantageous. We just know that they're more common because they're more activated. Okay. So can you give yourself points here? This would be 2 points and what is this? Question number 6, 6, 7, 8. These are 3 points question. Okay. So 1, 2, 3. Yeah. 6, 7, 8 is 3 points on the old test. There's no more 3 points questions on the new test. So 3 points, give yourself 1, 2, 3 points, please. Let's look at the next one. We got malaria. Malaria is a disease caused by eukaryotic parasites. All right, so it's not a bacteria. First of all, it says eukaryotic cells, so it's not a bacterial disease, okay? Malaria is caused by a protist. That mosquito is not a bacteria when it says eukaryotic parasite. The enzyme DOHDH is essential for the synthesis of pyrimidine bases, including thymine. So okay, good. So they tell you about this enzyme. What is so special about this enzyme? You need an enzyme to make bases, to reproduce. You have to make bases. So you can imagine without this enzyme, then you can't make these bases, then you cannot reproduce, right? So to synthesize these bases, this enzyme requires electrons, so to tell you more detail, yeah? It requires electrons that are donated by the electron transport chain. This protein called cytochrome B. So cytochrome B donate electron to this enzyme, yeah? And this enzyme will synthesize the bases needed for reproduction. All right, so we're told information about the enzyme. Furthermore, we're told that this drug, so you don't have to pronounce the drug, yeah? Drug irreversibly bind to cytochrome B and inhibits the growth. Okay, so the drug targets cytochrome B. If you block cytochrome B, there's no more electron transport, then the enzyme is not functional. If you are not functional, you cannot make these bases, so the parasite die. Does that make sense? So all this information is there, and you just have to retrieve them for your explanation. Yeah, let's look at part A. So based on the data, estimate the effective dose of the drug that caused 50% reduction. So yeah, you just look at the 50% reduction right here. 50% you go down. So if you use less than 10 to the negative 3, they survive. If you use 10 to the negative 3 molar, exactly your 50% growth. That will be the 50% reduction, right? Very close to 10 to the negative 3, yeah? Okay, so that's the answer, yeah? So 10 to the negative 3, all right? So 1 point for saying that. So this is number 7A. Again, 7 is 3-point question. So about 10 to the negative 3. Use the same unit, micromolar, okay? So you don't have to spell it out, just micromolar, all right? Second one, based on this, describe how the drug most likely suppressed. So we're told exactly how the drug works. It binds drug. Say no to drugs, but drugs here, okay, white people drugs, so they're legal. Okay, anyways, not very funny, but okay, a lot of drugs are illegal, but somehow if they're pharmaceutical company made, which is Wall Street made drugs, they're okay. So drugs are just bad in general, whether they help you or not. So try not to use drugs. Yeah, even these beneficial ones supposedly. There's always side effects. So drug blocks cytochrome B protein. That's what it says. I'm just repeating what it says and putting into a logical thought, right? So how it suppresses the growth? Huh? Cytochrome. Just sometimes these are just forever. Okay, I'm just gonna write it out. So cytochrome B, a block cytochrome B. Okay. Transfer, electron transfer to DHODH. So it blocks cytochrome B's electron transfer. Yeah, I guess a possessive here. It blocks cytochrome B's electron transfer to DHODH enzyme, right? Without functional DHODH, no nucleotides can be made for cell division, right? For growth and division, for example. I just tie them together, that's it. Okay, so that's 1 point. Describe, you cannot just put a one statement. You have to make sure you're describing what they want to see. So how the drug suppresses. So we just put together whatever we are told, okay? Identify cellular location where cytochrome B is likely found. Well, we know cytochrome B is electron transport chain, and it's a eukaryotic cell. So mitochondria, inner membrane, yeah? You can just say mitochondria, or the inner membrane of mitochondria, okay? So either one is fine. You don't have to be that specific. The location, right? What is the location? Inner membrane mitochondria is more specific, or simply mitochondria. So 3 points. Can you give yourself out of 3 points, please? Let's look at the last one. Okay, last one here, we have a phospholipid bilayer, and we have ABCDEF. So proposed model of hypothetical plasma membrane. Each model represents a small portion of a hypothetical plasma membrane. Illustrate the orientation of the phospholipid. So for number A, you should know that phospholipid bilayer, the polar head, this is polar, hydrophilic, they love water, and the tails are nonpolar, hydrophobic, they're afraid of water. The polar is the phosphate, and the glycerol is negatively charged. And the nonpolar part are the fatty acid. All right, so student proposed 6 different models of this hypothetical plasma membrane. Identify the model that best represents the plasma membrane of a hypothetical cell, so there's another cell, that exists in a non-aqueous environment, and whose cytosol is similar to that animal cell. So this is your hypothetical cell, okay? So a hypothetical cell. Inside is similar, the cytosol is similar to animal cell, that means aqueous. Aqueous means water filled, right? Aqueous means polar. And the cell exists in nonpolar. So non-aqueous means nonpolar. Aqueous means dissolving water, right? So that would be a nonpolar. So this is hypothetical. So if I want to make up a membrane for this hypothetical cell, I want the fatty tails against the nonpolar region, and I want the polar heads against the aqueous region, right? Okay, inside this aqueous, right? So I'm looking for which model that fits the best, okay? And inside is polar, outside is nonpolar. So model A doesn't really work that well, right? Because you have the cytosol is inside, and the non-aqueous environment, this is outside, right? So we want the polar inside, nonpolar outside. So polar inside, nonpolar outside would be this one, right? So outside is nonpolar, so fatty acids are nonpolar. Inside is polar, so the phospholipids are polar. So D would be the best answer. Okay. So that would be D. Okay. And provide two pieces of reasoning. So it's kind of stupid. It just said that phospholipid. Okay, so I'm gonna just write it out. Okay, so number 8, model D. Yeah, and your reasoning is that the aqueous cytosol interact favorably with the polar heads of the phospholipid. That's one. And the non-aqueous or the non-polar portion, the non-aqueous environment, okay, interact with the fatty acid tails of the membrane. Okay, so kind of like that. So they want two justification, right, for your reasoning. So D is the best. Okay, so that's it. So it's very easy. So membrane. Last year, the first question was on the membrane as well. You have to know that the phospholipid is polar and nonpolar. The fatty tails are nonpolar. The phosphates are polar. So you have to know that. Okay, so very important that you do. All right, can you please add up all the points for question 1 through 8? It is out of 41. So if it's incomplete, you didn't finish it, just let the office know. So let me post this one here, save it. And next week is what is this? Multiple choice again. Yeah. So do the multiple choice on the Google Doc, submit it, and then we have your score. Try to do as fast as you can on easier ones and save a lot of time for the multi-part questions that has the experiment and data. So again, this is 8 questions, but once again, we said that the new test, there's only 6 questions. The first 2 are 10 points, 8 to 10. And then 3, 4, 5, 6, 7 are 4 points each. Yeah. Okay. Alrighty, guys. Then I'll see you guys later. Thanks for coming in. Yeah. Okay. Bye, guys. Take care. See you guys next time. Bye.